I'm sorry for not being able to make it. I'm a little bit sick, but it's great to be here. Can you guys hear me?
Yes, we can hear you. And
Great, thanks.
I hope that you aren't feeling too bad. I know he has a flu, so he couldn't come. But,
Yeah.
but we will hear from him. And Andreas, where are you speaking from?
I'm at Stanford right now. I'm not far from you. I'm in
Okay,
Northern California.
okay. So Andreas is, okay, if I had to have one word for Andreas, it would be ambitious. He takes on the really hard problems. And he has tools. He has computational tools. He has experimental tools. And he also is going to take on a problem that goes against all of the received wisdom in the neuroscience community. Okay, the tradition in neuroscience is that you have to have control. You have to have control of the input. You have to have control of the output, because otherwise you won't be able to interpret the recordings. You won't be able to figure out what the cell is doing, right? And so the typical experiment, you have a monkey in front of a monitor, and you give a flash it with a visual stimulus, and the monkey is trained over months to be able to interpret it, match it, remember it, whatever the experiment is, but it basically is sensory, motor, reflex. Okay, and we've learned a lot from that. We've learned about how different parts of the brain, encode different types of sensory input and different types of motor output and so forth. But what we really need to do is what is going on in the brain when an animal is freely moving, making moment-by-moment decisions on what to do next, and, and what do you do with the data? If you could record from the neurons in the monkey, how are you going to interpret what you see? We now have mathematical tools to actually do it. We actually are able to do it. But maybe we'll hear from Andreas how he's going to do it. Okay, Andreas.
Thanks, Terry. So, this is a relatively easy task for us. Humans are these dogs and muffins. And, when we show them to people that have never seen this type of stimuli before, you can easily classify them after you look at them for a little bit. However, if you show this type of stimuli to even the state-of-the-art deep learning models by OpenAI, by Google, by Anthropic, and I just did a test this morning on OpenAI's o1 Pro version, which is their most advanced model, and it got 13 out of 32 correct. So it's basically performing at chance. And the key here of what is intelligence in the brain is generalization, the ability of brains to generalize to unseen situations. And although, deep learning and AI right now is really amazingly impressive in its ability to exhibit intelligent behavior, it still struggles with outside the training distribution. And big techs bet on this. It's nicely illustrated by this meme here, which says my model cannot generalize out of distribution. And then, Debbie says here, then we'll train it until there's nothing left out of distribution. So basically, if you can collect enough data and you have enough resources, we just heard yesterday the announcement of a $500 billion project to build a big supercomputer down in Texas to train these kind of models. This is where all the money is going. It's basically generate more data or get more data, build larger compute, and hopefully you'll be able to solve this problem. But of course, there are some issues with this. We know that we're running out of data. We only have one internet. And also, there is another example, the brain, that seems to be able to solve this problem much more efficiently and is capable of generalizing outside its training distribution quite well. So the holy grail in neuroscience in many ways is trying to understand this isomorphism between brains and the world. That is, what is the correspondences between brain activity and its structure and things like perception, action, cognition, memory, learning, and so on and so forth. And this is has been what we neuroscientists for more than 100 years now have been trying to solve. And this process at the what we call representational level, where we study neural activity, we abstract away from the details, the implementation level details. We just look at the activity patterns of individual neurons or populations of neurons, has led to many seminal discoveries in the field, from the discovery of the action potential by Adrian almost 100 years ago, studying essentially correspondences between the perception of force, in this case, I was how much force it was exerted on a muscle, to the activity of a neuron, and all the way down to the Moser's beautiful work in the entorhinal cortex, where they found these grid cells that are thought to be important for spatial navigation and the formation of spatial maps. But if you look at many of these discoveries and you talk to the people that make these discoveries, they were often serendipitous discoveries. They were not driven by a specific hypothesis about how the brain works. And as Terry said, they were in fairly restricted conditions, experimental conditions. For example, Adrian was actually, he isolated the nerve fiber and the muscle to study the stretch reflex. And if you talk to people like Charlie Gross that discovered face cells, it wasn't because he had a hypothesis that these neurons were encoding face cells, but that's because he couldn't drive them in any other way. So he started like showing his face in front of the animals or his hands and so on and so forth. So these discoveries were based basically on human intuition and human ingenuity, but not necessarily being discovered with a systematic or formal way of hypothesis-driven science. And this is very nicely illustrated by this 60-minute documentary by Hubel and Wiesel, where they describe how they discovered their seminal discovery. And here is David Hubel talking about the discovery in this film that you can see. And he was saying that they were using, because they were students of Kuffler, they were using the spots of light to try and understand how the visual cortex is driven. And one day we all heard the story. This light had some contrast, as you can see here, that created this edge. The neuron was driven by this edge, and then they discovered orientation selectivity that led to many, seminal discoveries in the field, and even inspired, to some extent, convolutional neural networks. So if we want to understand, for example, the macaque cortex or the macaque visual system, there is in the order of 3 dozen maybe, or dozens of visual areas. Each visual area has different cortical layers. Each area has many different cell types. So there is a lot of complexity. And if we want to study more complex systems, like we want to have, as Terry said, like animals just exploring naturally their world and interacting with each other, then we're faced with a really problem of unfathomed complexity. And it seems to me that this more traditional reductionist approach in neuroscience has yielded tremendous opportunities for discoveries in the last 100 years may not be powerful enough to take us to the next level. The problem, for example, in vision lies in the fact that if you have a monitor that, let's say, even if you just look at it in a very restricted way, but you have a monitor that is 256 by 256 black and white pixels, you can generate to the 65,000 possible patterns. So it's almost an infinitely large number of patterns. And this makes the problem of even just studying natural vision in front of a monitor, well, the animal is not even like roaming in the world or interacting with other animals and experiencing the complexity of its natural habitat, very, very difficult. And if you add this, as Terry again was saying, behavior, multimodal stuff, internal cognitive states, then it becomes very difficult to try and decipher the neural code, how is information encoded at this level of neural manifolds with different cell types and different areas and so on and so forth. So in the last 10 years or so, there are these two revolutions that I think that if we bring them together in neuroscience, it will be transformative in trying to understand neural computation. And one of it, and a lot of it thanks to the Brain Initiative, who very wisely invested in the first 10 years in neurotechnology. So the Brain Initiative spent a lot of its resources in neurotechnology, and it developed neurotechnologies now that when deployed can record data at very large scale, something that was not possible 15 or 20 years ago. I mean, there were a lot of amazing efforts, but now we have these things scaled up and more standardized. For example, now here is a microscope from Ali Pasha's Vaziri's lab at Rockefeller that can record close to 1 million neurons simultaneously with two-photon imaging. And here is the NeuroPixel technology developed at IMEC that now you can scale it up, and even in a behaving monkey, you can put 10 of these neuropixels and get thousands or through tens of thousands of neurons. So we have the ability to collect large-scale data. And on the other hand, we have deep learning, a revolution that has transformed, is transforming everything. They're sort of, started with, many, many years ago. Very few people believed that you could model intelligence using artificial neural networks. And again, Terry was one of the pioneers in this. But now we have these technologies that if you have a lot of data to train neural networks to make predictions, then they become very good at absorbing this data. So the idea here is to bring together large data collection and deep learning to try and create a model of the brain. And I will describe to you our approach here. We call it building digital twins of the brain and this concept of inception loops. And here is a bunch of papers. And in fact, I have to say here that Terry had the same idea in the 80s. I believe he collaborated with Bob Desimon and applied pretty much the same methods that I'm telling you now, but back then they just didn't have enough data to train these models very well. But the idea is pretty much the same. So this inception loop idea has basically four key components. The first is you record large-scale data under the ethological or natural conditions that you want to study the system. In this case, of course, it's still not fully natural, it's under constraints, but this is the beginning. So here is a mouse that is an awake behaving mouse. We do this also in monkeys that is viewing 5,000 unique natural images in a matter of a couple of hours. And then you record about 5,000 neurons. So now you can collect within 2 hours, and you can do this experiment many days in many mice, so you can collect hundreds of thousands of neurons. But in just 2 hours, you can collect 25 million training points to train a neural network. And the next step is you use deep learning. In this case, you use a deep learning architecture. And instead of training it to predict the next word like you have with ChatGPT or trying to predict the label, the class label of an image, you're just training it to predict neural activity given an arbitrary natural image. And then once you have this model, you have something that is quite remarkable, I think. If you have an accurate model or a digital twin of those neurons that you're trying to study, now in silico, you can explore it in ways that is unfathomable to have been done in the brain, in the original brain. For example, you could imagine that instead of running one experiment, you would parallelize this on a GPU and run what would have taken 1,000 years of experiment. You can run it in a few hours. Or in this case, you can do even something more fancy because this model is differentiable. You could backpropagate the gradient to and optimize things like finding the most exciting images that would drive this neuron, which is what, for example, Hugh Bonnet-Weasel tried to do in the visual cortex back in the 50s. So, and here is one of these experiments where you start with an arbitrary, with a white noise stimulus, and then you back propagate the gradient through, but instead of the same way you do back propagation, but instead of changing the synaptic weights of the neural network, you just change the input here, just the pixel intensities. And then when you optimize the images like this, you get these images on the left here called the most exciting images, or MEIs. And what you see here is that these MEIs, and this was a surprise to us, at least in the mouse visual cortex, they look very different from Gabor filters. This is on this here is the left rows here. They seem to encompass things like corners. Some look like checkerboard patterns. Others look like closed crescents. T things that look like T junctions. And when you search through a very large, like hundreds of thousands or even close to 1 million natural images, then you find these features occurring in these natural images. But of course, this is all discovered in the model. And the models, as we know, they're extrapolating maybe outside their training distribution. So they're hallucinating, right? They're not producing things that are reasonable. But because we can do these experiments in closed loop, then we can synthesize these images. And when we show them back to these neurons, they drive these neurons much higher than any other images we've tried. So this shows you that this method of using large-scale recordings, deep learning to build very accurately predictive models, and then doing in silico exploration, in this particular case, using the differentiable capabilities of these models, you can synthesize images that when you show them back, you get these highly exciting images. And when we did the same in monkey V1, we find that in monkey, the story is much closer to Hubel and Wiesel's Gabor filters. In monkey, you see a lot of these neurons having Gabor filter-like properties. So now we'll show you two examples. One is on bioRxiv, the other one is just fresh, is not published, that shows you novel discoveries that we can make based on this approach. And then I will end up with a more sort of futuristic view of where I think these type of methods could be heading. So one is studying contextual modulations. We know that perception is dependent on context. For example, as I showed you, if you look at these images here, in order to disambiguate and know if this is an eye or a raising, you need contextual information or the surround information. This is something that is very well known in vision and in other sensory systems, and is known as center-surround interactions or contextual modulation. And for many years, the classical dogma in the field of studying contextual modulations was either in the retina or in the primary visual cortex, and in some cases in areas like MT, where people used parametric stimuli, and in this case, oriented edges, for example, in V1. And the dogma here is that if you have a stimulus that is preferred in the center of the receptive field, in this case, let's say a vertical bar, if you show vertical bars in the surround that are congruent with respect to the center, then you get this massive suppression. So neurons that have their center and the surround being stimulated by a stimulus that is the preferred stimulus in the receptive field of the neuron, they get suppressed. And if you have the orthogonal pattern, you still get suppression, but it's less of a suppression. So this idea of center-surround interactions and was based on this type of experiments, and there is literally maybe hundreds, if not maybe 1,000 papers along these lines. However, if you look at a lot of these stimuli that optimally drive the center of the neurons, and here on the left, I'm showing you the stimuli in the mouse visual cortex that I've shown you earlier, and on the right, some stimuli in macaque area V4, of individual neurons in macaque area V4, you see that they like much more complex patterns that are not easily parameterized. So now the question becomes, how do we study center-surround interactions or contextual modulations when the stimuli are not easily parameterizable. And to do this, Jakun Fu and Catherine Franke in the lab did the following experiment. They built a model that was very similar to the previous one, but it also included a lot of stimuli that were masked. And this model was very good at predicting contextual modulations of arbitrary patterns, spatial patterns that you would show. And now once you have this model, you can do the following experiment. You can optimize for a specific neuron the most exciting, find the most exciting image, and then you can optimize this around. You can cut the gradient in the center and saying condition on these most exciting images, what is the image that will give me further activation? So you're allowed to now put contrast in the surround, not in the center, in the surround. And what we find to our surprise that we were getting these images that look like they were congruent or completing with respect to the center. We call this sort of pattern completion. And if you create stimuli that were maximally inhibiting, then it created this disruptive type of stimuli. And of course, you have to verify this. We showed this back to the animal and did closed loop verification. And then we quantified this pattern completion and disruption by measuring what's called the representational similarity. And I'll show you very briefly what we did. You take these natural images, so you take the most exciting images and you find in 1 million, let's say, natural images, patterns that resemble the most exciting image. And here you find this specific image, for example. And now you look at the surround, and now you compute the representational similarity, how similar is the excitatory surrounds with the natural images. And what we find is that the excitatory surrounds, that means the surrounds that were synthesized by the model to activate the neuron beyond its most exciting image in the center, were also more resembling natural images. And we did this also. This is not just a idiocracy of the mouse, it's also true in the monkey. We did this in the monkey, and what you see here, this is a bunch of most exciting images for the monkey. And when you create the surrounding images, you see that it forms things that look more like completing, whereas when you show here, sorry, the inhibitory ones, you get these disruptive ones. I think I'm missing a slide here. Okay. So the other thing that we did more recently is studying macaque area V4. And again, here you show these natural images. Here we use colored natural images, and you build these kind of models. And what we find here is that these are most exciting natural images that you can also do something more simple here. You can just filter through, you can show to these neurons 1 million natural images, something that you couldn't do in the neurons. You cannot show 1 million images to a neuron unless you do very fancy chronic recordings, but here you can do this very fast in these kind of models. And what you find here, these are 3 neurons, is that you see that the most exciting natural images here, these are actually rendered images, but they have naturalistic characteristics. They have some similarities between them. We call these diversely exciting images, but you see, for example, that the neuron in the top likes some red pattern in the upper part of the visual field, whereas the one in the middle likes some zebra stripe type of pattern, and the one at the bottom likes some sort of checkerboard pattern. So these images, they have some invariances, these neurons. They do not, it's not just one point in this tuning manifold, but it's like it's a ridge. And this ridge has images that have some similarities and some differences between them. In this case here, they share, for example, the same color, or these ones like these zebra stripes, and the other one likes the checkerboard. But what we found surprising to us is that a lot of these neurons are not super ultra sparse in their encoding. For example, there are a few images that are most exciting, as you see here by the activation distribution, but also there's a few that suppress the neuron. And if you look at these ones that are suppressive to the neuron, they're also very selective. We call these the least exciting images. So there is another group of images that, for example, for this neuron 1, it gets excited by these reddish objects, but it gets suppressed by this type of checkerboard or grid-like patterns and so on. So there is this tuning manifold that basically shows you that the way to think about this tuning function of these neurons, they have one region in feature space that is highly activating and one that is highly suppressive. And another neuron is like that. And what we find, I don't have the data to show you here, but when we analyze this thing, we find that these parts of the feature space are shared among the population. So there are specific image patterns that seems to be shared among the populations that are either highly exciting or highly suppressive. So these tuning functions do not span this entire feature space, at least for the stimuli. So this bipolar coding, we are still trying to understand what it means, but it could be related to enhancing information or by stretching the tuning function of these neurons. So to summarize, so far, this idea of collecting large-scale data, building models, and doing in silico exploration, and then testing these hypotheses that are generated or discoveries back to the brain, I think is a very powerful way of building models of the brain. And I think as we get much more multimodal data from different experiments, the way we are seeing in AI, these multimodal models where they're combining language with video, with audio, with other modalities, building these very powerful AI models. In neuroscience, we'll start basically building these foundation models of the brain where we can bring together, for example, different tasks, all the way to freely moving animals, neural activity, all the way down to transcriptomics. And the advantage of these models, and one of the models I'll show you now is very much like that, using a transformer architecture. They can be built in a way that you can stitch data from different modalities, different levels of analysis, even different species together into a single model. Here is one paper from our lab and another one from Richard Blake's lab, Eva Dyer, and the Lajoie lab in Montreal. And these foundation models, these multimodal foundation models, are basically, using many experiments, many mice. In our case, it was built on mouse. Right now, we're building one on monkey, where we get data from many experiments and we stitch them together to build this multimodal foundation model. And as I said, this version of the model is based on a transformer architecture, although other architectures, including recurrent convolutional neural networks, work equally well for this type of experiments. And what is important here is that these models now, because they're trained on so much data, they can generalize to new neurons and new experiments or new mice very fast. For example, here I'm showing you a model that we take a number of mice and we build a core foundation model. And then we take new data from a new mouse. And with 16 minutes of data from the new mouse, we can perform as good as if we were to record for 76 minutes from that same mouse and build a model of its own. So this shows you that as we're getting more and more data, we're starting to see the scaling laws that you see in AI apply here too, where we're getting more and more data. These models become more and more powerful, and they can feed new data much faster. These models are not just good at predicting the type of stimuli they were trained on. This particular model was trained on natural movies, But then when we showed it static images or even parametric stimuli like gabors or coherent random dots, it performed also extremely well, showing that it could generalize outside its training distribution. This model can also be used to decode because these are multimodal models that basically you can build them in a way, although this model wasn't exactly built like that, but you could still reconstruct it. These models, you can build them so you can, their input is also their output. So you could, for example, then use neural data. You can unmask the video and reconstruct the video from it. And what you see here is the reconstruction of the video. This is mouse visual cortex, so it's low resolution, but this is a novel video that we could reconstruct from this model. And then recently, in collaboration with the Allen Institute and Princeton, we did this microns project where we had from the same mouse both the function and the structure of these neurons, the anatomical information and large-scale connectomics, and the same model that I showed you earlier. These could predict the cell type, the morphological cell type, based on purely anatomical criteria as were defined by the Allen Institute. So this model was never trained on an anatomical criteria, but its functional characterization could predict fairly well the anatomical information for these neurons. So to close, I think that ultimately what we're interested in neuroscience is to understand this isomorphism or correspondence between brains and the world, right? Whether this is our perception, our action, our memories, as our social behavior, right? This is where we're trying to figure out these correspondences. And what I'm advocating for is now with our ability to have technologies that can be deployed at scale, and we have deep learning and AI, we can go through these coping exercises, first building these digital twins of the brain that enables us to do a much more extensive exploration of the correspondences between the world and this digital twin. And then we can close the loop by testing it back on the brain. So I see this thing as these three sort of modalities here: the brain and artificial neural networks, or in silico digital twins of the brain, or of any other AI systems, as a matter of fact, and then the world and making these correspondences. And then I would like to acknowledge the people in my lab that did the work and our funding agencies. Thank you.
Okay, we have enough time for several questions. Yuri, you want to step up to the microphone, please?
Andreas, in a conceptually similar experiment, Seiji Tanaka in Riken showed, I think, 1,000 pictures to a monkey, and the goal was to see generalization of similarities across images reflected by a single neuron. Now, of course, the background here was that the brain reflects the statistical probabilities of the environment and so on. It fit everything perfectly, except every now and then it showed that this is the MEI, the most effective image for these neurons, because it was the two eyes and so on. But the real most excitable image was a fractal. It has absolutely no similarity whatsoever to the 50 other pictures that were the best drivers. So the question is that, do you see some oddities like this? And what can we learn about these idiosyncrasies when it comes to vision recognition?
This is an excellent question. And I think I'll try and rephrase it, hopefully not paraphrase it. So one of the big questions in neuroscience, and actually it's true also in AI now, is how interpretable is the brain for humans, for us to understand it, starting at the level of the activity of individual neurons and then going to neural populations, right? And of course, we neuroscientists always believe or hope that it's interpretable, and we find that neurons, individual neurons or populations, encode latent variables in the world. And then, as Yuri said, there is these sort of experiments where people have gone and showed maybe hundreds of stimuli, and they found that it likes phases, but also a fractal. And then how do you make sense of it? And this supports the view that maybe the neural code is not interpretable, at least the way we're studying at the level of individual neurons or looking at the things that activate it mostly. So and this is something that is a very big open question in neuroscience, especially in visual neuroscience. If you go to areas like V4, or you go to areas like IT, and so on. What we find so far, and this is still preliminary because we're just starting to build these models for the higher visual areas, is that if you build these models and then your search space now becomes essentially unlimited, you can show it 1 million stimuli, then in our hands right now, everything becomes interpretable in a very weird way. Like these neurons that look like they were not interpretable, once you increase the space of the stimuli that you show them, and you truly find, let's say, the most activating images that are much more than these fractals or these toy things that Tanaka showed, then you see stuff that are very highly activating that looks interpretable. Now, this remains to be determined to what extent, and this is still preliminary, but right now we find that if you follow this approach and you build these predictive models, things look more interpretable. And this is also related a little bit to the Golden Gate Bridge example that Anthropic had with their model, where they find, some interpretability in deep neural networks too, where people thought it's all parallel distributed and things are much more messed up. But this is a really excellent question.
So there's another flip side to trying to decode, what the neuron likes is to create, and you point out that there's an almost infinite number of possible images, and the ones that actually exist that make sense to us, like this room or nature, is very, very small subset, right? But here's the additional discovery that is really shocking: diffusion models of image generation. So, I heard Eero Simoncelli, he spent his whole career trying to denoise images, right? And it turns out that the way that you can create, with DALL-E and some of these other diffusion models, photorealistic scenes just by giving it some instructions, like I want a room with a bunch of neuroscientists in it looking kind of bored. It will do that. How could it do that? So with diffusion models, what you do is you start out with images like the room, and you add some noise, right? And then you add a little bit more noise, and you're trying to make now predictions to go both forward, adding the noise, and then subtracting the noise, and you keep doing that until you get just something that looks like a noise, like a random pixels that you showed. But what's amazing is that literally, if you do that at the same time that you encode the sentence of what it is. Now, if you have the sentence there and you start with pure noise, it will reconstruct something like the room that you asked for. Photorealistically, in other words, it picks out a subset of measure 0 that somehow is associated with those words, and it's very well organized and structured, and you can now manipulate it. You can say, well, I need more people in the audience. There's enough people in the audience, and so it will add more people to the audience. I find that completely astonishing. It's just not something that, maybe Andreas, maybe it's something that you anticipated with all of the experiments you've done, right? You've created these very interesting, most exciting images for your cells. And so maybe what I think you are showing is that these are not random, they're structured, they're like little pieces of structure, right? And by the way, this is something that comes from, you were showing the the wavelets, the Gabor filters. So, it was shown, back in the 80s by Bruno Olshausen and also by Tony Bell in my lab, that if you just ask, with using independent components, what are the fundamental basis functions for natural images, they're Gabors. So it's not too surprising that the simple cells you see at the very beginning, it's a good low-dimensional basis function. It's sparse. So there's a couple of principles here. You want something that picks out from the infinite number of images what's the minimum number of little basis functions. But when you go up, it's for more complex things that you're trying to reproduce, the details, you need to have the kind of things that you are discovering. So, and by the way, have you ever gone to inferotemporal cortex with your method?
We soon, not yet, but that's where we want to go soon. We're very curious to go to all these high visual areas and beyond, also areas like the hippocampus and torhinal, perirhinal, to see.
Interesting.
Yeah.
And what about monkeys? Have you thought about monkeys?
Yeah, this is in monkey. I mean, we're doing it in monkeys now. It's all, most of the work right now, we're doing a lot. We continue the mouse work, but a lot of it is in monkeys. The stuff that I shown towards the end, that was monkeys before.
Aha. Okay, another question. Go ahead.
Excellent work. So my question is do you think this kind of neural network trained on animal data work generalized better than just artificial neural network? Because there's sort of inherent context encoded in the already in the data, let's say the animal behaviors or the activities. So take your dog and a muffin, for example, because you told me, right? You told us to distinguish the dogs from the muffins. So then I can react, I can distinguish these two objects better, classes better. But if you flash just let's do images randomly, right? So in a surprise me those two images, I might not distinguish those two objects immediately, I guess.
Yeah. So you're asking if these networks, because you're right that if we train these neural networks, we can also use them for computer vision. Will they be able to compete with AI systems that are trained just on behavioral data?
Yeah, maybe this kind of extra structures in the behaviors and activities encoding extra information rather than just the structures of.
That is a really excellent question, and we do not know that. I mean, we hope that will be the case. So, for example, these multimodal models, even the diffusion models that Terry was talking about with language and video or static images, if we add neural activity data to it too, will they improve? For example, will they have a better sample complexity? They'll train faster, or will they generalize outside their training distribution? Because they are representing, we're basically giving the model not just the labels, because what is AI now? It's basically trained on human behavior, right? That's the fuel of AI is human behavior, right? And of course, GPUs, right? It's like humans have written text over 2,000 years. We put it on the internet, it got trained. Humans have labeled images. They have written they have written captions for images, and that's how the diffusion models like DALL-E works to create images given a label. If we add to that mix neural data, will they improve? That's an open question. But there is some intuition or some evidence that they may improve. Because, for example, one of the things that we think that the brain has, it has this disentanglement of the latent variables in the world that may make it more powerful.
Thank you.
So, you showed those muffins. I have to say that if you stare at one of those muffins, it does begin to look like a dog.
Yeah, it's true.
And I've had, these moments. I remember I was hiking up in Glacier National Park, and they told us to watch out for bears. There are bears up there, and you have to be careful. And we were walking on the trail, and there's a bear. right ahead of us. And I said, everybody stop, stop. There's a bear. And it turned out to just be a stump that looked like a bear. In other words, it just takes a few features and, with foliage around it to fool your visual system. And maybe that's a good thing, that we can generalize beyond, to be able to see things, project into scenes things that aren't actually there, but actually could be there. I don't know. I was just, by the way, this is, when I was a graduate student at Princeton in physics, I spent a summer in Charlie Gross's lab. You had a slide up, and I've forgotten the date, 1984. He had a famous paper where he went, Hubel Wiesel spent their whole career in V1. He went up to IT, which is the other end of the spectrum, and reported that he saw these cells that looked like face cells that, so I was really curious about this, so I was going to spend a summer and actually see the real data, the recordings, and so forth. I was a little skeptical. And by the way, everybody was skeptical back there. Nobody believed that there are face cells. It was before your time. But, it was a little bit, it wasn't really, didn't seem likely, or it was just kind of, why faces, right? In any case, so an experiment would take 3 days. You prepare the monkey the first day, and then you start recording, and you keep recording literally for 3 days. And you would do one cell after the next, right, infratemporal cortex. It was very hard to drive the cells, very hard, unlike V1. But they had a big box full with all kinds of things, toys, and Tanaka did the same thing, by the way. I visited his lab. He had a different box. They were Japanese toys. And the stimulus that was the best one, for most of the cells, was a toilet brush. Kind of a circular wire with little brushes coming off. And they actually did it in that famous paper. They actually had a toilet brush up there as one of the stimuli. And then I said, it suddenly dawned on me that Charlie Gross and, could you go back to that slide, Andreas?
Yeah.
Okay, if you go back to that slide, the very beginning, there you go, you just passed it. Okay, there he is. Charlie had a beard. It turned out that 2 or 3 people in the lab all had beards. His was black at the time. This is a little bit later. And, Bob Desimone. That's Bob Desimone, by the way, above the IT faces. Bob was a graduate student in the lab at the time. And they all had these bushy beards that looked a lot like toilet brushes. Now, it's possible. In IT, we know it is the area of the visual system that can recognize and learn to recognize new types of objects and features and so forth. And so it could well be that after this poor monkey, after being in the cage and seeing all these guys in the lab with bushy, and actually, because they fed them, right, there was a positive reinforcement going on there that there are new cells in their infratemporal cortex that had these features. Okay, but one last question.
Hi, Andreas. Thanks for the talk. Very cool stuff. I was wondering. So you showed all these foundational models to train the transformers, the networks, with the neural data, and then make these experiments on the models. So it's like much faster, and you can do like that at scale. But the title of your talk is Less Artificial Intelligence, and I was just wondering, like, if you could say a few words about like the other direction of this thing, like making, what can we do with all of that?
Yeah, I mean, I can tell you a little bit about it because we're very interested in that. I didn't show much here, but so for example, you could, a lot of these models, as I have shown at the beginning, there's I'll talk about 2 areas. One is inference, like if you show, an image and you have to like ask questions about it. There is a tremendous amount of progress in AI in trying to do this, but there's still a lot of gaps, especially in new situations. Like, for example, the classical thing is with autonomous driving. If you haven't trained it a lot in under the conditions that you're testing it, it doesn't perform very well. So here the idea is that if you have these neural networks go through a neural bottleneck, they are less artificial in the sense that they're not just trained to do the tasks that, let's say with the labels that you get from the humans, but they're also trained to predict neural activity. Then when they're shown novel images, because they've disentangled these latent variables much better, they will be better at generalizing outside their training distribution. That's one of the areas that we're interested in. And it's tightly related to this thing called adversarial robustness. I don't know if you're familiar with it, where basically they're just more robust, these models. The other one has to do with video generation, although, this thing is becoming very good, but I don't know if some of you have played with things like Sora. If you look at Sora, for example, at least the Google version, they say is much better, but I haven't got access to it yet, so I don't know if Evo 2 maybe solve this problem, but it doesn't seem to have an understanding of physics and of things like occlusion and, for example, object permanence. Like, it will generate video that looks kind of nice, but if you look at it, it violates physics in a weird way. Like things that go behind something, they disappear, or cannot even do two balls hitting each other. So the idea is that these models that are trained on neural data from the brain of things like monkeys that have a very similar visual system to ours, they will be much better at this task, or they will need less data to be trained to be as good. That's the one area that we're interested.
Thank you.
Great. Okay, let's thank Andreas again. So, yeah, thanks very much for coming, and goodbye.
Thanks.
Okay, well, I think I'm supposed to give a summation, but it's already late, so it's going to be very short. This has been really, for me, a very, very informative day. All the talks had new things in it that I'd never seen before. People were showing, unpublished data, unpublished recordings, unpublished ideas. And what and Andreas, I think, really put it really well there at the end, that we're living through this amazing revolution that is occurring simultaneously, both in terms of the tools and techniques that we have developed from Brain Initiative, innovative neurotechnologies, for recording for more neurons and reconstructing brains at a much higher scale, and also manipulating. We saw from Mark's talk, not only can you record from enormous numbers of neurons and over larger and larger areas, but you can manipulate them. You can turn them on and off. You can use cell-specific. So that is a powerful way of not just correlational data, but being able to test for causality. If you really think that the PV cell is doing something, generating a particular type of wave or ripple or whatever, you could go and manipulate those cells and test it or activate them. At the same time, we have this amazing revolution in artificial intelligence. Who would have thought that, that you can get a network and you can talk to a network, right, that gives you answers to many different kinds of questions. I mean, it's in some ways it's already super intelligent in the sense that it has more knowledge base than any human ever existed, right? I mean, it has absorbed the world's knowledge. Now, it may be superficial, or you can complain that it doesn't know how to reason. Well, I know a lot of people like that, right? So I wouldn't compare it to Einstein. I would compare it to the average human. It does pretty well. Okay. So, but that's amazing. I mean, it really it's a mystery, and it's almost as mysterious as the brain. The difference is that it's completely accessible to us, and we can look at every single activity pattern of I showed it in my talk, every single activity pattern of every unit and every connection. it's totally open. And the only reason why you call it a black box is that you don't actually look into it, as I said. And we have mathematical tools for doing that, and Andreas showed what the tools were that he's using. So we're really, I think, at the very beginning of a whole new era that is leading toward going to be a much deeper understanding. And it's going to be a collaboration between these two areas now. We can create things that have amazing capabilities on one hand, and then we have this incredible brains, and we're able to look at large-scale patterns of activity and see things we've never seen before. And they're all, in a sense, in the same computational universe, right? They're all these large-scale, massively parallel units, highly interconnected. And learning is a very important part of modern AI. Learning is the secret sauce. And we now can create networks, with enough data that actually are able to learn to do all sorts of interesting cognitive things. And so, it's just beginning. And it's certainly the impact that this is going to have on the world is getting more and more real every day as more and more people now are using these tools. I mean, and we're talking about people in every area. They're using it for helping them write computer code, or write for architecture students for very quickly creating new structures and being able to use them for interesting new designs. And I mean, it's just like it goes on and on and on. And the beauty is that we're going to be able to understand both, I predict, because the mathematicians have gotten interested. And I once gave a talk to a bunch of mathematicians, and I said, look, these large language models are just equations. And, it's just a hairy equation. You can write it down. And who knows how to analyze equations better than a mathematician, right? So, they're the ones who are going to explain it to us, I think, ultimately. And the explanation isn't going to be in words. It's going to be in very high dimensional spaces. It's going to be with geometry. It's going to be with topology. It's going to have much more substance. And this is going to be the kind of explanation that physicists have given us for the universe, which is a much deeper and a much more sophisticated way of looking at complexity than I think what most people now out there are trying to look for in terms of, I want a simple explanation that I can write down and, make me happy. That's not the way I think that we have to really think much more analytically. We have to think much more in terms of maybe even new mathematics will have to be created. It should be interesting. And in fact, there's somebody in the audience who actually has already made progress along those dimensions. Would you stand up, please? No, she's looking around. There she is. Stand up. Yes, you. So tomorrow, Chan is going to give a talk in a graduate training, graduate course that I'm teaching on double descent. Does anybody know what double descent is? You do? Okay. So double descent is how the testing error, you have a training set, a test set, when all these are networks, you train with gradient descent. So when you start training it, as you give it more data, the test set performance gets better and better, right? But with a given amount of training data, as a network gets bigger, suddenly the test data goes up because you're overfitting. And this is what statisticians have been telling us. But if you don't stop, even if the training error goes to zero, if you don't stop, there's a transition where suddenly it starts coming down again. It's called double descent. over-parameterization, which is an anathema, right? The statisticians have been telling us, don't go there, don't go there. That's a bad place to be. You shouldn't have too many parameters, right? This is the way I was taught. But it's not true with these large language models, or, these transformers. And so Chan has discovered a mathematical theory based on physics with Nigel Goldenfeld, her thesis advisor, that she will tell us about tomorrow. So I'm really looking forward to this. You see, this is how it's happening. It's happening with young people who with better ideas than the old people. and the future is really bright. I really am looking forward to it. Okay, thanks everybody for coming. I had a great time. And Maxime, Maestro Maxime, stand up and take a bow. Take a bow. Take a bow. Okay. He did all the heavy lifting. And you notice that he has a beard. I think that might have something to do with his success. Okay, bye, everybody.